Hello, my name is Siobhan, and I am with Team Triple T, the Totally Tubular Teenagers from Skagit Valley College in Washington, and our project is on developing an eDNA isolation protocol detecting goldfish DNA in a controlled aquarium. And here's our instructor.
Hi, my name is Kristen Hoffber, and I'm a biology teacher and just one of the biology teachers leading this undergraduate research project. So it's my pleasure to introduce this research project. First, though, I would like to acknowledge all of the biology faculty who are involved in this project, and they're on the slide. And I would also like to acknowledge our community partners, Pacific mammal researchers, who also helped us develop this project. Finally, our project received some grant support from our own college and also the National Science Foundation. Okay, so the goal of the project this year was for student groups to develop a method to isolate environmental DNA. And to make this practical, we decided to use a fish tank as a model system. And the students were tasked with developing methods to isolate eDNA or environmental DNA from the fish tank and use PCR to detect goldfish environmental DNA. Now, this system was connected, or this model is connected to the larger question that our collaborators are interested in. And that question is, can we use environmental DNA to monitor harbor porpoise populations in the Salish Sea? So I'm going to pass this presentation back to the student group.
Thank you, Kristen. Hi, I'm Isabella Clement, and as you know, our research question for this project is, can we use environmental DNA to monitor the populations of harbor porpoises in the Salish Sea? While harbor porpoises are not a threatened species in the Salish Sea, their tendency to remain close to shore leaves them vulnerable to direct and indirect human impacts. Currently, Pacific Mammal Research uses photo ID to monitor harbor porpoises, but researcher error and limited time can negatively impact the documentation of all activity. This is where eDNA technology comes in. It's non-invasive and a reliable method. So what is eDNA? Simply put, environmental DNA is cells gathered from an organism's environment. After reviewing other research, we found that there is no standard method for eDNA isolation, so the focus of our research was to find the method that worked best for us. To develop a procedure, we used water samples from a goldfish tank and compared nylon and mixed cellulose filters in order to map out a procedure with the most efficiency for extracting.
All right, thank you again. So here is a picture of our whole poster, and I'm going to be focusing on some zoomed in areas of this poster. So like Isabella said, our goal was to test different membrane filters, different membrane filter pore sizes, and storage conditions to find the most effective solution for eDNA isolation. The first step in our process, which you can see here, we chose to pre-filter a 500 milliliter sample into two 250 milliliter parts using a common coffee filter in order to avoid blockage by algae and small debris. This was later changed to a 1 liter sample. After doing so, we used vacuum filtration here to capture any eDNA in the pre-filtered sample. So we ended up testing 0.2 micrometer, 0.425 micrometer nylon membranes, as well as 0.45 micrometer and 0.8 micrometer cellulose ester membranes, as well as a 75% ethanol solution in the freezer to protect from denaturing due to UV light or warmth. All right, so after filtration, we extracted eDNA from the membranes using physical and chemical processes with the DNEZ Power Soil Pro Kit. And we then ran PCR amplification of the fish CO1 gene. While waiting for this to be completed, we also made an agarose gel using a mold to create wells for our samples, which during a later lab time would be filled with the PCR products along with UV dye to give us a visual confirmation of our procedure's success or failure, which Lauren will be talking about in the next section.
Right, thank you, Siobhan. So total, we ran through the filtration process twice and completed 3 gels to visualize the product. The first gel shows in lanes 2, 3, and 4 no amplified DNA products on trials with deionized water, concluding that our handling resulted in no DNA contamination. Lanes 5 and 6 look at amplified fish DNA samples on the 0.45 micrometer nylon filters. There's still no bands to show to collect a DNA product. So in our second gel, we move on to retest these samples as well as a positive fish DNA to see where the source of the issue arises. Because there's successful amplification of the positive control and still unsuccessful amplification of our DNA samples, we conclude that this problem was within our samples. We think that they either sat too long between filtration and PCR runs, or that the water samples were not large enough to have a DNA product to amplify. The final gel tests larger water samples of 750 milliliters and 1 liter within a week of the initial filtration. Our DNA shows successful amplification here at 750 base pairs with the 0.45 micrometer nylon and 0.45 microliter MCE filters in lanes 2 and 3 showing the brightest bands. The 0.8 micrometer MCE filter in lane 5 showed no amplification, so we conclude that this pore size was too large to catch any eDNA. Based on these results, our finalized methods are thus. Filtration process should be preceded by a pre-filtration with coffee filters to remove algae and dirt from an unclean seawater sample. Our filter water sample should be filtered through 0.45 micrometer filters and stored in a 75% ethanol solution in a dark freezer for no more than a week to prevent DNA deterioration. During isolation, 360 microliters of solution C1 should be used before centrifuging to create enough supernatant to extract the DNA from the filter. This procedure was developed using Goldfish DNA, so the question remains if the 0.45 micrometer pore size is the best universally, or whether the Hovæbuhr porpoise cells will be different and present problems to require new filtration testing.
Thank you again to everybody who made this project possible. It's been a pleasure, and we're excited to see where this research takes us in the future.